Okay. Right now, we can also look a little bit of the easy way to remember these things. So heterozygous 9331 ratio. But in Mendel genetic dihybrid cross, if we change genotype, so we could get different phenotypes. So one is this. So still we keep in the heterozygous combination, one heterozygous, and other one change, capital A, if we take, so phenotypes are 12 to 4. So we try to remember, then other than 9331 ratio, there is a 12 to 4 ratios. So always we make quick exercise when we are giving the ratios like this. We try to see whether it is coming with the Mendel total ratio, the ratio's total number. So 16 is a number. So we try to see whether it's matching with 16. Yes, we can remember, in addition to 9331 ratio, there's a 12 to 4 ratios. If we have a heterozygous, another one phenotype change to a homozygous form. So capital A, capital A. This is one example. We get 12 to 4 ratio. And we can also get ratios like this. Second ratios, 6622 or 1133. So in that case, we keep the heterozygous combination like this, and we are changing here simple B, simple B. So it's this time we take homozygous recessive. So if we take homozygous recessive in the other parent, we get 6622 ratios. I think it's for time management, in case Mendel genetic disease asks, so 12 to 4, 6622 ratios, we can remember like this. So if we know this in our mind, okay, these ratios come in this combination, it's easy. So these are the two combinations we should remember. 12 to 4. In that case, we change other parents to a homozygous dominant. One phenotype change to homozygous dominant. The other one phenotype change to a genotype change to homozygous recessive. Then it's a 6:6:2:1 ratio is coming. Probably it's easy if you know these things. If similar question is asked with different ratios, we can quickly figure it out answer, so we can save the time. Then other one, so here capital A, simple B, simple B, capital A, simple A, simple B, simple B, and here capital A, simple B, simple B. So we take both the other side, homozygous dominant and homozygous recessive. So if we take homozygous dominant and homozygous recessive, third combinations are ratios are 1 to 1. 1 to this is 1 to 1. So we have 9, 3, 3, 1 ratio, 12 to 4 ratio, 3 to 1 to 1 ratio, 1 to 1 ratios. 1, 2, 3, 4. So we could get 4 different type of phenotype ratios with the dihybrid cross if we change the genotype of one parent, other one keeping heterozygous. So this might be useful to remember in case similar questions come with different ratios. we can save the time, quickly find the answer. So example, 2011 question. So is it clear?
Clear, sir.
Yeah, I think you make a small note. I will upload these videos. So you can make a small note somewhere like quick revision like things. Okay, these are the ratios we can get if we change this genotype of dihybrid cross in Mendel genetics. Okay, next question. Again, 2009. In a plant species, fruits can be either red or yellow. Both these type can be obtained as a pure line when two plants with yellow fruits were crossed. So yellow crossed with the F1 plant, two plants with yellow fruit were crossed, yellow and yellow. All F1 plants produce red fruits. So F1 generation red. When F1 plants were inbred, that is red and red, progeny had 27 plants producing red fruits and 21 plants producing yellow fruit. So 27 red, so 21 yellow. Which of the following statement is incorrect with regard to this inheritance? Okay, first of all, the 27 and 21. Do you find these ratios are fitting to Mendel genetics?
No? No.
I think Mendel genetics, we learn at least 1 to 1 is coming. Here, 1 to 1 is coming, 8 and 8. If we cross both homozygous conditions with heterozygous. Uh, so here, they are not even fit into 1 to 1 ratios because 27 is very high from 21, so it doesn't fit. And the other one is we also feel that how come when you cross yellow and yellow, it get red one, and when you cross red and red one, they get red and yellow one. And also this is a one phenotype. It's not dihybrid, it's a one phenotype. So if Mendel genetic there is a one phenotype, so it could be a it is like a capital R, simple R, capital R, simple R, like this combination we have to get because it's only monohybrid cross. See, because one red and yellow it's only, and we have no idea that which is dominant and which is recessive like things. All it says can be obtained as a pure line. Both these type can be obtained as a pure line. So uh, if red one is pure and the yellow one is pure, if we cross this, then we get a different phenotypes or different ratios, but it's not mentioned here. So it says red, red. So first one they take red, and they take the red one, red, red, and they get a yellow now.
So yellow, red, no, sir?
Yeah, yellow, red. Yes, yellow. So when they get the yellow and yellow, F1 is red. Then F1 cross with red, yellow and red. So all does not fit into Mendel genetic, right?
Yeah.
Okay, color of the fruits could be produced by the interaction of two genes. Gene interaction. Second one, epistasis. Okay, epistasis is similar to, again, gene interaction, epistasis. They are going very close to each other. Dominant allele of two genes may be necessary to produce red color. All plants with red fruits of F2 progeny could be pure line. All plants with yellow fruits of F2 progeny may not be pure line. Now here, if we use all the knowledge we gain from genetics, uh, what do you think, which one may be most incorrect, goes to the incorrect side?
So third answer.
Today is difficult for me to use this pen. I had to correct the problem, I think. Okay, we learn if the flower is yellow color in Mendel genetics, if it is comes like a capital A, and this is yellow, we take a capital Y, capital Y, it gives a yellow color. They are pure lines, so they always produce pure line, because originally we learn in Mendel what he did this purple color and white color. The purple color flowers always produce purple color, and this generation keep it as a pure line. I mean, if it is giving white color, white color, simple Y, these are always keep as simple Y, and they are pure line. So in the first experiment, he used pure lines. As a result, he got all 100% flowers are heterozygous with the purple color. Now, in this case, F1 generation, and this is the parents. And then when he go to the second one, so he got a F2 generations. So now if you look, this one you can consider as a pure breed. But this one is not. Did you understand that?
Yeah.
So if we go for F1 and F2 generations, we can't observe pure lines. No, so if they say F2 generation, all plants with red fruits of F2 progeny could be pure lines. So we have a small doubt in this case. If they mixed up red and yellow here, So how could be the pure line again? F2 progeny. So now we can understand if it is Mendel genetics, non-Mendel genetics. So this could be some kind of a gene interaction. Could be an example of epistasis. And we have a 3, 4, 5. All plants with yellow fruits of F2 progeny may not be the pure line. Now, if you take 4 and 5, there is a little bit of issues coming. One say it should be pure light. Other one say it is not a pure. It's not pure light. Red is pure. Yellow is not pure. So one is opposite of the other. So fourth and fifth one is something telling us in the same choices this is this, but the other one say no, it is not this. And also when we have a F1 F2 breeding experiment, it's hard again to obtain the pure lines because of the mixing of these alleles. So we can take fourth one as the answer. All plants with red fruits of F2, they are not could be a pure line because it's incorrect. It might be incorrect. There is a very less chance of having pure lines when we are crossing many generations. So this question is a little bit not fit into one of our all the concepts. So we need a bit of uh, all information to handle this 2009 this question. It is not a straightforward from a theory. Like previous one, yes, uh, there are certain instances we had in the past like this.
Uh, so why can't the answer be third answers?
Hmm?
Why can't the answer be three, sir?
Dominant alleles of two genes may be necessary to produce a red color. So you are saying it's incorrect.
Yeah, because it can be simple as well, no, for one.
Dominant allele of two genes may be necessary to produce a red color. So uh, if we see the like say yellow color, now when you have a we write like this, yellow color is like this. Yellow color, color, capital, okay, yellow. And capital C, simple Y. And if we cross this one, capital C, capital Y, capital C, simple Y. So why give the yellow color? So Y and R. So I write here because it's not clear. So Y, C, C, R. So if we cross C, Y, C, R. So codominance and incomplete dominance like things, we can also get something. So this is yellow and yellow. So when we cross this one, so C, Y, we have a CR. So we have a CY, Y and R. Now Y and R can give the red color. Right? So here, I mean, if we say here is a capital Y and simple R. So if we have a Y, Y, YR, then RR. I mean, just I'm getting example. We can get these combinations YY, YR, and RR. As it is gene interactions, and this is example of epistasis or gene interactions. So in dominance, codominance like conditions. So we can also assume that two alleles required to make the red color. Because how do we explain then if we have a two yellow color cross, other one is give red color, and two red color cross, we get 27 and 21. So it is something we have to understand. These two genes should work together to decide these colors. One cannot completely make the color. If one is making the color, it is again going to the Mendel genetic. So when they are combining to make the colors, it is gene interaction. So when we accept color of the fruit could be produced by interaction of the two genes, this is epistasis. So we have learned actually under epistasis, the gene interaction, there should be some kind of combination necessary to express the color. And top of that, when you take all the five choices, all plants with fruit of F2 progeny could be pure line. So this is easily we can understandable than any other choices. When you have F1 and F2 progenies, they are again mixing up. So when they're mixing up, it's hard to understand whether they can become again pure line. Pure line is they express only one character. So if it is yellow, then it's only yellow. Yellow, yellow, it's get yellow. But here, yellow, yellow mix, they can get a red one as well. So this is something not 100% fitting to our all theories learned. So we have to use a bit of understanding of all these things to get the answer. 2013. So would you like to try this one?
Yes.
Oh, no, I think just okay. I think we will. You can uh, do this at home, right? I can send this one. This one. Okay. It takes time, so you can do this one. So we have done similar questions, I think, for this case. We have learned something similar concept to answer this question. So that is easy that you can come up with the answer.
So answer is third answer. No,
Third one.
no, sir. Fourth answer.
Yeah, I think we have done a very similar question before. Um, Only that question they have given additional guideline. These alleles are in different chromosomes. So capital T and simple T is one chromosome, capital Y and simple Y in another chromosomes. That was given in the previous one. So best thing is quickly, I mean, we have to also consider time management. If you have this kind of thinking questions, not directly from the theoretical part, so we have to exercise a little bit, and we also look how we save the time. So this 296 and the other one is 104. So being large numbers, I can write 300 and 100. So the rounded up to 300 to 100, that is the 3 to 1. 3 to 1 mean Mendel understanding, if you could quickly understand, it should be 12 to 4. So this is the ratio then. So then it's very easy. We know what is this ratios is coming. Capital T, simple T, capital Y, simple Y, and then capital T, simple capital Y, simple Y. So we remember 12 to 4 ratio is coming, heterozygous, and one is a dominant homozygous, other side. Yes, answer is right. So we have a method now for these kind of questions. So when you look at the questions, guess quickly the ratios, how to get 16. Like if it is coming to 12 to 4 or 1 to 1 or 8 to 8, 6 6 3 3, like that, 8 to 8, 9 3 3 1. Okay, this is Mendel genetics. And these are the ratio, the genotype should be crossed. So we can save time for these kind of questions. Here again, I take the same thing which explained. So we use this theory, this strategy to find the answer. Okay, 2013, getting a little bit easy now compared to the past 2005 or 6. Genetic questions become a little bit easy. So can you try this one?
Okay, sir. Okay.
Oh.
Uh, Fourth answer.
Woman has blood group B. So B mean it should be I, B, and the child coming up with O. Then IO is coming. Then there should be IO. So woman blood group is IB and IO, and identical twin sister should have the same blood group then. And then they marry a man with IA and IB. So this is IA, IB situation. So then we get I, this one with this A, B. This one with this IB, I, B. And IA, IO, IB, IO. So AB, B, A, B. So A, blood group A, blood group B, blood group AB. Fourth one is correct, yes. Two 2014. Can you try this as well?
Okay, sir. Uh, Fourth answer, sir.
Yeah, this is a dihybrid test cross, right? So dihybrid test cross, we have learned previous, for the very beginning, we have learned that is, if you don't know this genotype, so we're going to cross with homozygous recessive plant. That could be simple R, simple R, simple L, simple L. And in that case, we have this uh, ratios like a 50-50, like this will very close to each other, like a 50% and 50%, half. So then we have learned what are the all possible combination. I don't know whether I have it here. It's also a bit just to better to remember it. I think. it was not here, I think the other document. Yes. Uh, red seed and long, red seed and long pot 130, red seed and short pots 145. Then both have a red seed, so it should be a capital R, and long and short, simple L, simple I. Yes, fourth one is correct. Because red, both red is there, RR, and the other one both having a long and short, it could be heterozygous then, 2014. So you can see now when we are doing 15, 16, 17, 18, 19, they are more streamlined questions and they are fitting to a concept we learn. So it is getting easy and easy, the genetic question with the latest papers. So we have like, if you look 17 and 18 like questions, they are quite very easily we can write answers. Okay, just to summarize now what we did so far on the genetics, we have covered Mendel genetics and non-Mendel genetics. That is the main part of the genetics because almost all questions asked, they come under Mendel and non-Mendel genetics. We can continuously do this question while we are learning before we're going to end the sessions because next topic is the population genetics, Hardy-Weinberg equilibrium. After that, breeding techniques. Breeding techniques are just concept and just like a theories, not much easily you can understand. We don't want much time to spend for the breeding techniques. That means soon we're going to finish genetics. In few classes, we'll finish unit 6 by doing all the questions. So if any question left or we do redo question, we can again do the same question maybe. Then we did all these questions in the past as well. I will send you the notes, right, for the rest of the lessons. Uh, then I will upload these videos as well. So you can check this, right, on the website.
Yes.
Okay, then we'll meet again next week, right?
Okay.
Okay.